Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Process in the Product, a new analytical tool for AAV production optimization and monitoring. I'm Caitlin Crouch, and I'll be moderating today's webinar. And joining me today are Blaj Bakalar and Ivana Petrovich-Koshmak, who will discuss challenges in AIV analysis and manufacturing, showcasing rapid capsid analysis, raw materials screening, transfection optimization, and workflow integration to improve efficiency and product quality. After our presentation today, we'll have a live Q&A session with our speakers, so please do feel free to pose your questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to them during today's session. Now, I'd like to introduce today's presenters. Blaj holds a BSc in biochemistry and a PhD in nucleic acid biophysics, and leverages his expertise as the PATfix product manager at Sartorius BIA Separations, integrating hardware, software, and analytical insights. Our second speaker for today is Ivana, who is the head of upstream process development at Sartorius BIA Separations, where she leverages her molecular biology expertise to optimize vector production and support advanced analytics for research and customer projects. So, without any further ado, I'm going to hand it over to Blaj to kick us off with today's presentation.
Thanks, Caitlin, and welcome everybody to today's presentation, which is going to discuss how process is the product when it comes to AAV production and optimization, where we're going to explore a new analytical tool to optimize AAV upstream production. Before that, let's do a little introduction into the AAV or adeno-associated viral vector therapy um, um, as well. So AAV is the most widely used viral vector in the world today and can deliver genetic material to a broad range of cells and achieve long-term, so stable expression. Interestingly, it comes in a lot of flavors, which are serotypes, plus recombinant, chimeric, and engineered capsids. This, as we'll see later, can make it a bit of a challenge. And but what it's good at is that it has a very low level of triggering an immune response compared to other viral vectors, and is most widely used also in the early gene therapy pipelines, where it's mostly used for orphan and rare diseases as well as single gene disorders. So here at Sartorius, we like to help drive these genetic therapy adoptions by helping customers address key challenges, which for today's AAV therapies are to further improve patient safety, reduce time to market for pipelines, and reduce costs per dose. So when it comes to addressing those challenges, low upstream yield is one of the bigger ones, where especially challenging can be also the low full percent capsid that can be produced in some of the upstream. And as well, difficulty obtaining highly pure and potent final drug product, long development times, and scale-up challenges during transition to larger volumes. So what we have been focusing on is providing tools that can help increase titers as well as yield during upstream, improving recovery of AAV capsids, full capsids with high full ratios, and shorten those development timelines with the use of DOE and analytical approaches, where design and to design an optimized scalable process. Now, if we take a look at analytics, which is one of the major tools that helps develop both upstream and downstream, because without analytical insights, it's pretty much impossible to know or optimize the process in any sort of way. There's a lot of methods that are available for analytics, and especially popular are the ones that are often utilized for quality control testing, as an example given here on the right. And when it comes to the challenges that I've shown on the previous slide, focusing on quantity and purity uh, helps resolve most of the challenges in the capsid production in the upstream. Now, these analytics are often used only after DSP capture steps that require sample purification because of their accuracy and robustness, um, which makes it a little bit of a challenge to use these analytics also in upstream. But when it comes to in-process analytics, you don't need to necessarily rely on like QC tested analytics because the goal of in-process analytics is to have process understanding and control. Because at the end of the day, with AAV production, the process is the product. So if we look at the process, most of the interesting things start happening post transfection, shown here as a little plasmid. So after we transfect the cells, AAV production actually starts. And optimizing this step, both the transfection as well as the production window, can help not just increase the yields of full capsids, but also increase purity even in the upstream. Because if we can make sure that we don't produce as many product-related impurities, such as empties, we can ensure that recoveries of full AAV capsids in this downstream can be more robust. So challenges to achieve this is to have a good screening and DOE throughput and good consistency of the process, as well as have it be scalable. In order to address this, we've developed a novel tool, which we call the PATfix AAV Switcher for upstream analysis of AAV production. So looking at the PATfix Switcher, the first thing to note is that compared to traditional analytics, it's actually a 2D chromatographic analytical system, where on the first step, we capture the capsids from the sample and then do the empty full separation in the second step. When it comes to sample preparation, all that is required is to dilute the sample with buffer, adjust the pH to binding conditions, as well as centrifuge or filter the sample from the upstream, at which point it can be applied to the system and a reliable empty full readout can be achieved. We equip this system with a lot of detectors because we're analyzing challenging samples. So we include mouse, fluorescence, and UV detectors. The analysis itself takes about 25 minutes per sample. And all of this is controlled by a single software that both runs the system as well as helps analyze the data. And within the software itself, a calibration curve can also be uploaded of an AAV standard or included, which then helps quantify the capsids, both empties and fulls, further. So if we then look a little bit closer on what's happening with the chromatograms as the samples are being processed on the first and the second column, I would like to draw your attention here to the box on the left, where you can see that on the sim trap column, we actually captured the capsids and then elute them with a very rapid change in pH. And you can see dissolution of the capsids happening by the orange and black signal, which increase as the capsids are eluted. This allows us to elute in low salt concentration and allows us to also do an inline dilution to then bind these eluted capsids to the second column, where you can see here at the middle of the slide where the empty-full separation then happens. What we've recently done is we've increased the resolution between the empty and full peaks, and we've been developing this method and system for quite a while now on a variety of samples to make sure that it's a robust approach. And what we've been able to achieve is actually quite significant, even for such complex samples as harvests from upstream are. We're able to achieve very good empty full resolution with the newer method, which is shown here on the right, compared to the old method shown here on the left. With the resolution higher of 2, that means that at least for the empty and full capsids, we can achieve baseline separation. When the samples are also not too dirty or too complex, other species can potentially be observed besides the empty and full capsids themselves. However, this is, I would say, not a guarantee and shouldn't be and isn't quantifiable at the moment. To test how the system performs for challenging samples where we might have a large excess of either empties or fulls and to have still a good, reliable readout of the actual ratios of the two capsids, what we've done is we've tested the system with injecting 2 purified AAV samples, one that was 90% full capsids and one that was 90% empty capsids. In both cases, the recovery that we observed in the chromatogram closely matched the expected ratio between the full and empty capsids, as you can see in the table in the middle. And on the right, you can see the detector responses either from the empty capsids or from the full capsids. Empty capsid sample being in the dotted lines and full capsid sample being in the solid lines. Further, we've also tested how the system performs with repeated injections of such complex samples. So we've done column lifetime evaluation for both of the columns used. And we've concluded that for the first column, so the column that sees the dirtiest sample, we've done over 100 injections of the AAV harvest sample, which all had at least 1 mg/ml of the protein, per injection, and haven't observed any loss of performance from it. On the other hand, for the CIMAC QAHR column, which is our high reproducibility column and does the empty full separation, we've observed that the column can roughly last for about 20 injections of the AAV harvest sample, after which peak fronting for empties and fulls can be observed, lower resolution between empty and fulls can be observed, as well as potential elution shifts. But the good news here is that when it comes to monitoring an AAV process, as Ivana will be showing, usually more than 20 samples don't need to be analyzed per batch. So to actually hear how this tool can be used to do meaningful things and increase the titers and full percentages of AAV capsids, I'm handing over to my colleague Ivana.
Thank you, Blaž. So we're now going to the second part of presentation where we are going to have a look into different applications. So our upstream process development team faced a challenge when we started developing an upstream process. And that challenge was the lack of suitable, reliable analytical tool to measure empty-full ratio. And I'm sure that many of you following will also agree you had similar challenges. So for that purpose, we were very happy that our colleagues have developed AAV Switcher, and we decided to test it for different applications. Those applications being screening of raw materials, some optimization experiments, and later I will show process monitoring and also comparisons of some batches that we have produced. So first of all, we used PATfix AAV Switcher to test and compare different raw materials. So far, we tested it both on adherent as well as suspension cell lines. We tested it on media, on cell pellet, as well as harvest, the whole harvest, lysed. And we also screened different media. We screened for different plasmids, including helper plasmids. And we also tested throughout the different scales. So what this slide is showing is an example of one of our screenings where we tested 3 different helper plasmids. So on the graph on the left, you can see dPCR titer as well as percent of full coming from PATfix AAV Switcher. On the right side, you can see graph with chromatograms coming from fluorescence and light scattering detectors. From this simple screening, it was clear to us that the best choice was the helper plasmid number 2. And I'm going to show some more data done with this plasmid. So after screening, we proceeded with 2 rounds of optimization, which we did in design of experiment style. Here I'm only showing our data on titer and the percent of full without heat maps. But basically, what we have done, we have tested different cell densities, quantity of plasmid DNA per million of cells, as well as the ratio between total plasmid DNA and Factovir AAV transfection reagent ratio. And as you can see on the graph on the right side, in most cases, we observed either higher titers combined with lower percent of full, or vice versa, higher percent of full with lower titers, meaning that we would have to choose between the two. However, we also saw 2 conditions here under number 1 and number 5, which showed both nice titer as well as percent of full, the only difference between them being actually cell density. So without, let's say, detailed insight into our DOE results, which we did in mod software, you can just see by eye that these 2 are likely going to be the best candidates. This allowed us also to reduce the quantity of plasmid DNA that we used per million of cells in our previous process to about 0.75 microgram per million of cells, which is 25% less than what we had in our old process. So it was not only optimizing for higher titer and percent of full, but also helping us reduce costs. So with these conditions in place, we proceeded with the next round of optimization, which was looking more closely into ratio between RepCap, helper, and gene of interest plasmids. Now, these results were, these optimizations were actually performed with AAV8 capsid, which contained GFP as a gene of interest. So this is, let's say, a model system that we used. So just by eye, you can notice on the right graph, the condition number 10 showed high titer and high percent of full as well. So this condition was roughly a plasmid ratio of 1 to 1 to 2. And this was the condition combined with the previous results that we proceeded on in scale up. So we have scaled this process through various scales, the biggest one being 10 liters. And these 10 liters of harvest AAV8 material we used as an input for our downstream. Our downstream typically consists of a pre-capture step, which includes clarification and TFF, followed by 2 chromatographic steps. The first one being performed on Simultus SO3, which is a cation exchange, and then polishing done on the anion exchange, Simultus QAHR, as Blaž already mentioned. So on the graph on the left side, you can see an example chromatogram of our polishing step done with the traditional empty-full separation methods, where we have a certain overlap between the empty and the full peak, here marked in E2 versus E3. So in this process, we have achieved a significant reduction of total proteins as well as total DNA, corresponding in 1.4 logs for proteins and over 3 logs for total DNA. And during the polishing, we were also able to reduce the content of empty capsids to about 10%. The next example that I would like to give you is how we used PATfix AAV Switcher together with other analytical methods to scale up our upstream process. So this was our old process before we incorporated Factovir AAV transfection reagent and helper plasmid previously mentioned. And we have first performed a certain series of experiments on shake flasks, of course, and then we went on to the Amber 250 as our smallest available bioreactor scale in our lab. And we proceeded on to scale it to 5 liters, which at the moment when we were doing these experiments was our biggest scale. So what you can see in this table are data for titer as well as the percent of full between these 3 different scales. And what we noticed immediately is that our bioreactor scales were let's say, more comparable compared to our shake flask, which is also expected, having in mind that in bioreactor we are able to have control over more parameters, such as dissolved oxygen, mixing, etc., etc. So this old process we have now scaled up to 5 liters, and then we got a 10-liter vessel, so we proceeded on with 10 liters. But the next challenge was to determine what is the proper time to make a harvest. So we first performed an experiment on 5-liter scale that you can see on the graph on your left side. So what we did is we collected samples throughout the entire process, and we performed dPCR analytics, and we used PATfix AAV Switcher to determine not only full capsids, but also empty capsids. So empty capsids being shown in black, while full are shown in yellow. So what we saw in that experiment is that our process seems to reach a plateau of full capsid somewhere around 48 hours when observed with PATfix AAV Switcher. Our PCR was, however, giving slightly different data, but having repeated this process a few times, sometimes collecting also different time points, we have noticed that it is more consistent in our experiments that plateau is truly reached somewhere around 2 days post-transfection. So to look more into detail in this region of interest, we have also scaled up our process to 10 liters, and we have more closely mapped the process in between 42 and 51 hours post-transfection. So this is the part which is marked in a yellow box on the left side. We now made a zoom in on a higher scale. And as we expected, there were no significant changes in titer as well as percent of full. So we have, during this experiment, we have defined the optimal window for harvesting our AAV8 GFP harvest, which allowed us to first spend less time producing this material, and second, reduce potential impurities, because the longer we leave harvest in bioreactor, cells keep dividing, and we can have a potential increase of total DNA as well as total protein content. The next example I would like to show you, and this is the final example for today's webinar, is how we used this tool to follow our upstream process robustness. So you are now observing an example of our old process on 5-liter as well on the 10-liter scale. The corresponding tables are shown below of 3 harvests performed using the same protocol on 5, and then the identical protocol just on a higher 10-liter scale. So what we can see looking at this data is that on both scales we had a certain variability, but seems like the variability was reduced on our 10-liter scale, where we also introduced some smaller changes and fine-tunings of the process. It is also very helpful to have the visual on how these empty and full peaks look like on chromatography, because this is at the end going to be your main downstream tool to capture and polish AAV. So with this kind of information, we approached our downstream team who processed the material as shown before. So using Simultus SO3 as a capture and then Simultus QA-HR as polishing. And with our improvements in upstream process, now going back to the new process, so from old to new process, using the identical downstream protocols, we were able to achieve more than 2.6-fold increase in yield. So if this was not our AAV8 GFP model system, but if it was some gene therapy product, this would mean over 2.6 over 2.6 fold increase in the number of doses. And this all while keeping over 80% of full AAV8 particles, which, as I've shown you, we use the traditional QA separation method, which means that frequently we would need to make a choice between the yield and the purity. But by improving upstream process, we were able to achieve better results after downstream. This kind of process has reduced our costs, and if this was a gene therapy product, it would reduce cost per dose. It is also reducing the time to market, and it is helping us to improve safety of this product for patients. I would like to close up my part with contributions and acknowledgements. I have to say that all of the work that we have shown and a lot more that we are not showing in this webinar is a work of several departments. So departments of for viruses, department for pDNA and mRNA, as well as department for analytical method development. I would like to thank Blaj and Ajbe, who are our product managers, and also our colleagues from Sartorius Polyplus, Paul, Hakima, and Nadia, who helped us optimize Factovir conditions. And here I would like to close my presentation and hand over to Caitlin.
Great. Thank you so much, Blaj and Ivana, for those great presentations. We're now going to move over to the Q&A portion of our live event. So Blaj and Ivana, if you both want to join me on screen with your cameras and your mics on. Fantastic. I can see you there, Blaj. So I'm going to dive straight in. And the first question here is for you, Ivana. What dilutions did you use for the samples from your DOE? Sometimes titers can be even 1 log apart. Do you dilute every sample differently?
That's an excellent question. So the entire idea for using PATfix AAV Switcher for process development is that you have to make minimal tweaks. So in our case, we have diluted all samples equally, because we knew approximately after the first screening which titers we are expecting, which was in a range of E10, E11 vector genomes per ml. So our choice was to dilute samples eightfold. Of course, if you know already that you will have higher concentrations, you might choose to dilute your samples more. Or if you know that you have lower concentrations, you can also dilute your sample less. But then this might let's say, have an impact on the amount of impurities that you're bringing to the first column. And let's say it will not affect the chromatogram on the second column, but you might be able to focus on using the light scattering detector quantification.
Brilliant. Thank you so much for that in-depth answer, Ivana. Our next question here is also for you. This audience member asks, upstream samples tend to be dirty. How does this affect the results and column lifetime?
Right. So as I mentioned in my previous answer, the whole point is that this system is suitable for analyzing very dirty, very complex samples. So this being said, we tested samples of all various levels of let's say, impurities, different cell densities, different media, etc., etc. So in our hands, the level of impurity did not impact the empty full ratio at the end. Also, if we would dilute sample in different ways, we would still get the same reliable empty full ratio. However, there could be an impact on the column capacity. So, as Blaj mentioned, when we tested multiple injections on the first column, we reached about, well, actually more than 100 injections of one sample. And for the second column, we reached over 20. So if you are diluting your sample less, you're bringing more impurities on the column. The more you're able to dilute your sample, the less impurities you're bringing, and the longer your column is going to last.
Brilliant. Thank you so much for that detailed answer. Just a reminder to our audience, we're getting lots of lovely questions coming in. We'll try to get to as many as we can during the live session today. But of course, any we don't get to, we will reply to by email. So please do keep sending them in. Our next question here is for you, Blaj. This audience member asks, what is the real throughput of this analytical chromatography? How much time would it take to analyze 10 samples?
That's a good question. So for 10 samples, it usually takes around 25 minutes to analyze one sample. So for 10 samples, it would take around 4 hours to do the analysis. So when it comes to high throughput, like compared to 96 well-plate solutions, this isn't as fast, but the system can easily run overnight. However, that being said, the main advantage is that you're getting, when you're analyzing one sample during monitoring scale-up, for instance, or something like that, you're getting answers pretty quickly on the current conditions in your bioreactor. So yeah. By the way, can you see me on camera?
Yeah, we can see you perfectly, Blaj. You're
Perfect.
crystal clear for me.
Thank you.
Fantastic. Brilliant. Thank you so much for that answer. Our next question here is directed to you, Ivana. This audience member asks, so in sample preparation, you can reduce pH to acidic. How long can samples stay that way before being analyzed? Do you have any data on how prolonged exposure to low pH affects the reliability?
Yes, that's an excellent point. So as Blaj mentioned, the first column on our system is a SIM trap. And then we have the shift from acidic to higher pH in order to elute bound AAV from the first column. So yes, we have to reduce the pH of the sample during the sample preparation. However, we have tested sample preparation and we left these samples overnight in the line, let's say on the machine, and they were tested one by one. So the system is, in that sense, automated. And we wanted to have a system that is allowing us to analyze all the samples, for example, coming from one DOE. So we're speaking in a range of, let's say, up to 220 samples. And we didn't notice any effect on the empty full ratio. I think our colleagues have also tried to acidify and leave sample for up to 2 days on room temperature and analyze the sample. And as far as we know, there was also no effect on the numbers obtained after the second column. However, more testing would be needed, and I would strongly suggest anybody who would like to test this method to test the stability of their own AAV. As mentioned, we produce AAV8 GFP in hex suspension cells. So if your sample is different than ours, you also might want to test the stability. But in our hands, overnight works without any problems and likely up to 2 days without any problems as well.
Great, fantastic. Thank you for that information. A really nice, thorough answer. Our next question here asks, can you analyze engineered capsids based on AAV8 and or produce in a different cell line, such as F89? And this audience member asked this question specifically for you, Blaj.
Oh, okay. So analyzing engineered capsids based on AAV8 in general shouldn't be a problem. Because usually when you're engineering capsids, you're not for the most part, changing a lot of the protein sequence, the capsid protein sequence. However, of course, as with anything, we are separating these capsids based on differences in surface charge, which means that if you're substituting, for instance, negative or positively charged amino acid residues, additionally testing to make sure that the capsids come within the salt gradient range would definitely be a good way to go. However, for the most part, from the samples that we've tested, it hasn't been a problem so far. We've also actually tested not just on HEK cells, but on SF9 producing so insect cells. And we've observed that when it comes to using the same construct as with for the HEK cells, we didn't observe major differences between the two.
Great, thank you so much for that answer. Our next question here has two parts. So the first part asks, can you use crude lysate with PATfix platform, or do you need to purify samples before analysis? And then the second part asked, what purification method do you use for that purpose? Blaj, Ivan, I'm not sure which one of you want to take a go at answering this first.
I think I can take this one, and then Blaž, maybe you can add something.
Chime in. Sure.
This is a beautiful question. So the entire idea behind this analytical method is that you can use unpurified samples. So that being said, the way we prepare samples is if we're, for example, using the whole harvest, or if we're working with the cell pellet, we would lyse them using our preferred lysis method. And we tested different. We tested freeze thaw, hypertonic lysis. We also tested different lysis reagents. So after lysis, we would need, as I mentioned, to drop pH, and then also based on the conductivity of the media, or let's say if you're using salt lysis, you would have to dilute your sample significantly. So the sample is being diluted with the first buffer that is used for this chromatographic analysis. And afterwards, we're making clarification. Clarification is done either by centrifuge or combination of centrifuge and 045 PS filter, or just the filter, depending, let's say, how complex the samples are. If we're using media, there is no need to complicate things. If we're using, for example, cell lysate, we would prefer centrifuging and then filtering. And this is all the preparation that is needed. From that point onwards, the sample is simply placed inside the vial, and vial is placed on a tray that goes into the instrument itself, and that is all. So there are no other purification methods, no other protocols to concentrate AAV or anything else. So directly from bioreactor, lysis if needed, and then dilution and acidification. And after that, the sample is good to go.
Yeah, that's very well put. I don't think I have much to add besides maybe that we do provide a guide on how to do this as well. So that makes adoption much easier for customers.
Brilliant.
Yes.
Thank you both for that answer. Ivana, do you have anything else to add there?
Yes, I just remember that I probably showed in my slides. So we have also combined the sample preparation with another tool that allows us to automatize it further. So to avoid pipetting of multiple samples, which is called the Streamlink. So this instrument is helping with the sample preparation and also filtration step before going to PATfix AAV Switcher. But if there is anybody following us today who would be interested to learn more, please feel free to reach out.
Thank you so much.
For the Streamlink application, definitely. Yeah, that's a nice one.
Brilliant. So there you heard it, audience. If you have any more questions along that nature, feel free to reach out to Ivana or Blaj, or send in a question. We'll get to it offline if we don't get to it today. So thank you both for that really lovely detailed answer. Our next question is for you as well, Ivana. This audience member asks, when is the earliest you usually measure with PATfix Switcher? When would you start observing the empty and full capsids?
That's a good one. So if we think about it in a in a certain way. So we are transfecting cells. And then in the first hours after transfection, we have transcription and obviously translation of proteins. And then the capsid assembly starts taking place. So depending whether we are interested to observe the whole harvest or maybe we just want to have a look into what is secreted in media, you might want to choose different time points. So in our experiments, in our system, the earliest we've seen some significant quantity of capsids was approximately 12 hours. But nothing really interesting happens at that time point. So usually you will have your AAV production and also start of secretion. And then I would say the most interesting time is somewhere between 2 and 3 days post-transfection, when you, depending on your process, you might be able to see some shift in production of empty versus full particles. So I would say you can go as early as 12 hours, maybe 18 hours, as I've shown on one of the graphs. But I would say the most interesting part is between day 2 and day 3. And depending on time points that you're choosing, I would say at certain times there are no significant changes between the time points. So you might as well collect 1 or 2. Whether if you want to map some region when there is, let's say, more strong wave of AAV production, you can collect samples more frequently, maybe every half of hour or so, depending on your process.
Brilliant. Thank you so much for that answer, Ivana. Well, as you heard here, science favors the patient. So next question here is for you, Blaj. This audience member, following on from the last one, asks, is the PATfix switch available and for which serotypes?
Okay, yes. So currently, the system isn't fully launched. However, for anybody who is interested, for early adopters to adopt it, for instance, for either higher throughput analysis or for monitoring, do let us know either here in the chat or afterwards via email, and we can connect and discuss details. Currently, the system was developed on using the AAV8 EGFP sample capsid. However, that doesn't mean that it doesn't work for other capsids. It just might require specific fine tuning depending on the serotypes, which we are already doing for many other serotypes. It's just when it comes to launching it and showing the system performance, we've decided to go with AAV8 for now.
Brilliant. Thank you for providing that information. Sort of following on from that last question, this audience member asks, do you sell any instruments or do you also offer analysis of samples as a service? If yes, what is your turnaround time?
Okay. So we definitely focused more on system sales. However, we understand, of course, that CapEx purchases can sometimes be difficult. That's why we also offer analysis of samples for anybody who is interested. And usually the way we do it is, so you would send us your material and we would, after receiving it, it takes about 2 to 3 weeks to do the full analysis, which usually consists, first of all, if we're working with the material for the first time, we will do some quick sample analysis to see what we're dealing with exactly. So some test chromatograms as well as some other analytics, followed by a full suite of analytics to determine more precisely the data that can be extracted from the samples themselves. So that's why it usually takes about 2 to 3 weeks, depending on how many samples we get, to provide the not just the analysis, but also then the full report and results to potentially interested parties.
Great, fantastic. That's some really good information. So audience members, of course, if you have any further questions or you'd like more information on that, you're welcome to reach out to the team at Sartorius BIA and to Blaj and Ivan, and they'll definitely be able to assist you. Unfortunately, that is all we have time for today. As I mentioned, any questions we didn't get to today, we will reply to by email. The webinar will also be available from tomorrow. So please do look out for an email from us with that link. I'd also like to take this opportunity to invite you to take a short survey to provide us with some feedback on today's webinar. This link can either be found in the resources tab in your auditorium or you can look out for it in the email you receive from us tomorrow. So, of course, all that's left to do is to, of course, thank Blaj and Ivana for fantastic presentations, and thank you for answering all those lovely questions. And, of course, thank you to the audience for listening. We hope you'll join us again soon.
Thank you, Caitlin.